So hey guys, how is it going? And in this video, we are going to cover the topic of biological oxidation, and that is actually a small topic, but may cover up to 2 to 3 lectures, I guess. But I'm not sure about how many lectures it covers, but we'll we shall try to keep it in 2 or 3, I guess. Um, so biological oxidation, we'll learn about redox pair and redox potential, what is biological oxidation, what is electron transport chain, what is oxidative phosphorylation, inhibitors and uncouplers of biological oxidation, then enzymes involved in biological oxidation, and all this shall be covered. So let's start. What is biological oxidation, by the way, and what do you mean by biological oxidation? So you all know that you eat food. Why do you eat food? You eat food to gain energy, right? So what does the food do that gives you energy? Does it be like a battery and just inserts and gives you energy? No, it's not like that, I guess. So what does it do that gives you the energy? So that's the reason you're basically eating food, while some are eating for taste, I guess. But this main function is getting energy. Taste is a secondary function, yes, right? So let's say you eat carbs, you eat let's say proteins, uh, and you eat fats, right? These three are the main portion of your diet, not doubt. Diet actually it is, right? So what is the first step that you assume if you to get energy? Right, digestion. Okay, I guessed it. The first step is digestion. So by digestion you are breaking it pretty down into its similar compounds, like carbs break down into glucose, proteins break down into amino acids, and fats break down into fatty acids and glycerols, right? So let us write them down. Glucose, amino acids, fatty acids, and glycerol, right? So these three compounds, similar compounds, we get from digestion of the above respective three compounds, right? So what do you expect from this all now to be going into? Now, as eventually the first step of digestion has occurred, the compounds have become simple and ready to be absorbed or ready to be converted into any other compound. What do you think can happen? Either they can get absorbed, they can get transported to various places, either they can get reutilized and all. But let us assume the case of glucose alone. What does glucose do? After digesting, glucose basically gets absorbed by the cell, right? It gets converted into pyruvate via the glycolysis cycle, and then through link reaction it forms what? Yes, acetyl coenzyme A, right? So after that they form a common compound, that is acetyl coenzyme A, right? Now what does this acetyl coenzyme A does? It goes into the Krebs cycle, the TCA. So it goes into the TCA cycle, right? And forms what? Does it just go to the TCA cycle just to enjoy? No, of course not. It forms NADH and FADH 2, right? So when it forms NADH and FADH 2, what do the NADH and FADH 2 do that gives you energy? Well, they undergo some reactions, I guess. Yes, right. But what is this step? This is the second step, that is called intermediary metabolism, right? um, just let's go here, and yes, so this is called intermediary metabolism, right? Now what do this NADH and FADH do? They basically go into the, yes, electron transport chain and form what? Our energy currency, that is the ATP, right? So they basically do what is they go to the electron transport chain, do something that we will discuss in the upcoming segments, and produce ATP, right? Simple as that. So what is the third stage called? This is called cellular respiration, right? So now we have gone through the digestion, we have formed simple compounds. The simple compounds have then formed acetyl coenzyme A. Then the acetyl coenzyme A, going through the Krebs cycle, has formed NADH and FADH2, right? Then that NADH and FADH2 went via the electron transport chain and produced ATP. So one more concept that I need to teach you right now, as it is important and you probably don't know it. Have you ever wondered why NADH and FADH2 are produced? Means you understand the question that if NADH and FADH2 both provide ATP, both are useful in providing ATPs, right? So why do we need two different molecules? Why can't we just work with NADH either or with only FADH either? Why? So the answer lies in here is very simple, that NADH gives you approx 2.5 ATPs and FADH2 gives you 1.5 ATPs. Now, something that gives you less ATPs will be produced from a less energy source compound, right? So, whenever a molecule that produces NADH or FADH, it is called a high energy compound. So, basically, giving an example. So, let us take an example of succinate. So, succinate is a high energy compound, but can it reduce NAD to NADH? No, it cannot. So what will it do? It will go with the cheaper version, that is FADH2. It will reduce FAD to FADH2, and then FADH2 will be guided along the electron transport chain to produce ATPs. So being processed or being made by a low energy compound relatively will give low ATPs at the end. Alright, as simple as that. So basically this was the concept that if the compound can reduce NAD, it will reduce it and form NADH and use it in the electron transport chain and give more ATPs. But if the compound cannot reduce NAD to NADH, it will reduce FAD to FADH2 using a cheaper version and producing less ATPs, but at least producing an energy, right? So basically this was how the three stages of biological oxidation occur, and what you eat gives energy. But now we'll discuss this in some pretty detail, as we are not just any students and we are MBBS students. We have been meant to know this all things, right? So going with the first topic, that is the redox potential and the redox pair. Right? So what do you mean by redox? Basically, first of all, redox by the name itself suggests, redox. So basically reduction and oxidation. Any reaction that has simultaneous reduction and simultaneous oxidation is known as a redox reaction, right? Reduction of one, oxidation of the other. So let us see it. An example. Let us assume A is a compound. It gets converted to AH. That means accepting electron or proton. Basically, we call it reduction, right? And let's just assume there's a compound B. But now, as we know, reduction of one, oxidation of other. And as this rule shall follow, so B has to get oxidized. But B is not in a reduced form to get oxidized. You have to be in a reduced form to get oxidized. You can only achieve everything when you have nothing, right? So let's add BH and guide it through the reaction forming B. Now what this happened is oxidation. So we can now interpret that A accepted an proton or an electron from B and converted itself to AH, that is its reduced form, and B donated the H or the proton and converted it to B, so in its oxidized form. So this is an example of redox reaction. So now this A and B are redox pair as they are undergoing a redox reaction simultaneously. And now what is redox potential? Basically, when you relate everything with examples, you have better clarity of the concept, right? So for the clarity, we'll take another example, that is of beggar and riches. So assume there is beggar and there is a rich person. So what do you think? The beggar will donate the money to rich person, or the rich person will donate the money to the beggar? Obviously the second cause, not the first, as who is called a beggar, by the way. The one who does not have money, a place to live, and begs for money is a beggar, right? So basically, in case of this compounds, in case of biochemistry, we call it redox potential. What is the potential, right? So basically, when there is a tendency to accept an electron and there is a tendency to donate an electron, right? So when you accept an electron, you are the beggar, but when you donate the electron, you are the rich. So we can surely divide this into two categories: positive redox potential and negative redox potential, right? So what is basically positive and negative redox potential suggest? When you have a tendency to lose electron, you get a negative redox potential. When you have a tendency to get oxidized, right? When you lose electron, you get oxidized. So you have a tendency to lose electron, you have negative redox potential. Okay. Now, what is positive redox potential? That is basically the tendency to, yes, of course. Very great. Tendency to accept electron or basically get reduced, right? So let us take some examples regarding this too. First of all, regarding this, have you seen NADH and FADH in most of reactions get oxidized to FAD and NAD, right? So NADH and FADH2, this both mostly have the tendency to get oxidized, means NADH to NAD and FADH to FAD, right? So you can surely count them in the examples of negative redox potential. Then comes the positive redox potential. Look, we are going to study the electron transport chain and its every single constituent in a pretty much detail. So I just want you to get aware of the names. I just want you to get aware of the names that are going to be studying in the final upcoming segments. So basically cytochrome B, cytochrome C1, cytochrome C, cytochrome A, cytochrome A3, and obviously coenzyme Q and half O2. This all basically love to take electrons. They are the beggars. They love to take electrons and will just take it whenever it comes in their way, right? So this was about redox pair and redox potential. Now we shall move to the electron transport chain, right? So yes. So what is basically an electron transport chain? By the name we guess. There is a transport chain of electron, right? Electron are being transported in chain. See, most of the science and MBBS is all about the words. You get the words, you get the concept, right? 80% the words reveal, 20% is on you, what you study. So let us first think about which reaction happens where. For example, when I talk about glycolysis, where does it happen? Let's create a clean separate area. So glycolysis, where does it happen? Yes, obviously in the cell cytoplasm. Yes. Then when we talk about the Krebs cycle, where does that happen? Krebs cycle or the TCA cycle, right? Tricarboxylic acid cycle. Where does that happen? Yes, in the mitochondrial matrix. Right? Now we are talking about the electron transport chain. Where do you think this will happen? Any guesses? Means let us draw a diagram to represent how this happens. We shall use, okay, let's use this color for mitochondria and this. So this is a mitochondria, right? This is the outer mitochondrial membrane. This inner mitochondrial membrane, this is the intramembranous space, and this is the matrix of mitochondria, right? So basically what we told is that glycolysis happens in the area that is cytoplasm of cell. Then that pyruvate through link reaction forms acetyl coenzyme, and that acetyl coenzyme A is transported in the matrix of mitochondria, right? And here we have the Krebs cycle. So when the Krebs cycle happens, it forms NADH and FADH that are required for electron transport chain. Means why they are required? Because they are high energy electron carriers, right? Means the high energy compound from which they form, like succinate, forms FADH. So succinate does what is that he's a great businessman. He likes to invest in small businesses and make them grow, right? So succinate just gives one high energy electron to FAD and reduces it to FADH2, right? Due to this reduction, the FIDH2 that is formed is now an high energy electron carrier, right? Now it will carry the electron and bring it from the matrix to the inner mitochondrial membrane where our electron transport chain works, right? So let us demonstrate it here. The inner mitochondrial membrane contains the electron transport chain that has the complex 1, 2, 3, 4 and complex 5, that is the ATP synthase. Right? Now ATP synthase is also called the complex 5. It's just what I told. When you want to know about a concept, just read its name, get what it means to say ATP synthesis. So ATP is synthesized, right? So basically this is a site where the ATP will be synthesized. We'll know this in just a moment. Just give me a moment and I'll make sure this concept is clear and we'll move to the main process. And I bet you will love it. Right? So outer mitochondrial membrane contains acetyl transferase and just one more thing before we move further here. Have you ever known in the condition of starvation, first the glucose from your blood is utilized, then the glycogen storage of liver, then the glycogen storage of muscle, then the body comes to starvation, right? So what will be utilized in starvation as there is no glucose, or in condition of diabetes, like where insulin is inefficient and there is enough glucose in the blood, I guess, so there is enough glucose in the blood, but the diabetes, the insulin is inefficient to pump the glucose into the cells, right? The cells need something for producing energy. We saw here that in matrix, the Krebs cycle, NADH and FADH2 are produced and in the cytoplasm glycolysis occurs. But if you don't have glucose in the cytoplasm, what will the cell use? So basically it's using fatty acids and glycerols for production of ATP directly through a process called beta oxidation. We shall cover this in the future videos, but it just struck my mind and I thought let's explain it first. So where were we? Yes. Now where were we? Yes, the glucose is inefficient, and we discussed how beta oxidation helps in producing ATPs through fats, right? But let us move on further and discuss what we are on to, and it was the ETC, right? So basically it has 4 complexes, complex 1, 2, 3, 4 and 5. The 5th one is ATP synthase, right? Now when the NADH and FADH are produced in the matrix of mitochondria, it gets transported to where? It gets transported to the inner mitochondrial membrane, and there it goes in the electron transport chain and just does something very incredible, and you will love it. I am telling you, will love it. If you understand it, of course. So it does something very incredible there and produces ATP, right? So now we know that the electron transport chain happens where? In the matrix of mitochondria, right? No, in the inner membrane of mitochondria. Sorry for the delay. I'm kind of a bit sleepy right now. So Yes, the inner mitochondrial membrane, right? Now let us take a look at ETC closely. First of all, we'll draw a diagram that will be best representing the ETC. So let's start. We shall use different colors. More enjoyment, right? So this is the ETC presented on the inner membrane of mitochondria, right? Now let's name this. This is complex 1, this is complex 2, sorry, this is complex 3, and this is complex 4. Now, what do we need to know? There is NADH, there is FADH2, right? So, let's first see the flow of electrons. What is the flow of electrons? That from which complex to which complex electron will be transferred. So, from complex 1 to complex 3, and from complex 2 to also complex 3, and complex 3 to complex 4, right? So, now, does the electron directly go from complex 1 to complex 3 like floating in the midair? But no, that's not the way it is. Here, have you ever taken a rickshaw or a bus? Of course, you would have if you are not just the son of a millionaire or billionaire. You would have taken a rickshaw and a bus, right? So, what does a rickshaw or a bus do? It just takes passenger from one place, transport them to other, right? We have a rickshaw man here and here. Sometimes you need to switch between two rickshaws or two buses, right, to go to a different destination far away, right? So these are two our rickshawalas. Without them, electron can't grow from complex 1 to complex 3 or complex 2 to complex 3 because it is a common pathway, because complex 1 and 2 are just neighbors and they have one rickshaw stand only, whereas complex 3 colony has different rickshaw stand and complex 4 is the final destination. You might be understanding the logic now, right? I guess. So if we go with the logic, we shall go with the magic, and the magic is we shall now see all the complexes with a bit of detail. And I guess the video is getting somewhat longer, so we'll stop by just discussing the names of each and every complex and what it constitutes of, and then we'll leave the rest for the next video, right? Let's start. So basically, in complex 1 you have sorry, NADH. Now the NADH that came from the matrix will give the high energy electrons, means it will transport the high energy electrons to FMN. FMN will transport it to FES, and FES will transport it to coenzyme Q, right? And now here, this NADH, FMN and FES, that's a boy complex 1. And this, wait, I'll relate at the last. So, succinate in the Krebs cycle forms FADH2, that means it is forming FADH2 means it is transporting its electron to FAD, that is being converted to FADH2. So basically it is transporting, right? Succinate transport electron, high energy electron, so FADH2, and FADH2 now transport it to FES, and FES transport it to coenzyme Q. So this FES succinate FADH2, this is our girl complex 2, right? Now coenzyme Q takes the electrons that are transported from both complex 1 and complex 2 to cytochrome B. Cytochrome B takes it to FES. By the way, the FES of the complex 3 is a little bit different, as it's also called Reisky. There is a basic change in the amino acids. We'll discuss in when we are discussing each and every part of the complex. Let's leave it to the next part for now. So basically FES transport the electron to cytochrome C1. Now cytochrome C1 will transport it to cytochrome C. So this thing here, cytochrome B, FES, cytochrome C1 is our boy complex 3. Now cytochrome C transfers the electron to cytochrome A. Cytochrome A transfers it to cytochrome A3. Cytochrome A3 transfers it to half O2. Now can you remember these all compound names that we just discussed in the positive redox potential? So then just apply the logic. If you just reduce the oxygen, half oxygen, what do you get? You get water, right? Transfer of electron means it is accepting an electron, it is transferring to somebody, and it is coming back to its original form. We'll just see it in a moment. So what do you think this is? Wait, sorry, just a second. That's a boy complex 4, right? So have you been correlating this diagram with this one? Looks a little same, right? What are we missing here? We are missing our two rickshawalas, this and this. What else are we missing here that we also missed in this previous diagram? And that is basically this little fellow, half O2. Why? Because as you know, other all places the electrons are being transported from one to another, but it goes as a full stop at last in half O2. When it is transferred to half O2, it basically gets stored at water. So this is the final electron acceptor, right? And so yes, this is the final electron acceptor, and it accepts the electron, converts to water, and becomes a permanent resident. Others all are just transporting, exporting, importing, right? So these two rickshawalas, why can they move? Why? The question is, can't the electron directly grow from here to here? But it doesn't. Why? Because these are immobile structures, complex 1, complex 2, complex 3, complex 4, right? But these rickshawalas are mobile structures. This coenzyme Q is of lipid nature and hence is movable, but This cytochrome C is of protein nature, and hence is also a mobile structure, right? Now let us get the basic idea of what are we going to study next in this topic. This is basically how this function, right? So ever thought of the glutathione mechanism? Glutathione basically does what? It accepts the electron from the free radicals or the single electron species and gets reduced. Getting the free electron is reducing the harmful effects of the free radicals or the single electron species and prevent them from damaging the cell membrane or any other structure. So basically glutathione sacrifices itself, gets a bullet in the chest and gets reduced. That bullet is the electron. But now next time there are any free radicals, there are any single electron species, what does stop it then? For that we need something, right? For that we have glutathione again. And from where do this glutathione come again? Basically you operate the bullet out, stitches, and you come back to normal, right? It was a little illogical explanation, but I think you got what I mean to say, right? Basically you have done something, right? Then you have to convert it back to its original form so that it can be used in the next cycle or in the next chain or in the next reaction, right? So this is what happens in this also. When NADH is given to FMN, FMN giving to FES, NADH gets oxidized, FMN gets reduced, FMN again gets oxidized, and in this oxidation process it reduces, because one oxidizes, one reduces, right? It reduces another. We'll discuss this in detail, but leave the headache for now onwards to the next video. And yes, how does this generate ATP? Let's get a concept overview. So you've basically seen a dam, I guess. It looks something like this, not a particular dam, but let us imagine it is. Now this is all the water. Here is a pressure, right? Here water will flow through this channel. It will push this due to its pressure, and the wind not wind, but the water turbine will rotate, and by rotating this there is a principle called converting mechanical energy into electrical energy and converting electrical into mechanical. There are two types of conversions, right? So this basically converts mechanical to electrical energy using something, I guess it is called dynamo or something like that. Well, not exactly, I do remember, but it was something like that. So basically this is what happens. Now same to same, our mitochondria inner membrane has also something like this process going on. First of all, we need this water. We need this water gradient to develop such that it can flow with pressure. For that, when the electron transport from this to this side, this to this side, this to this side, and this all happens, there is a generation of some calories energy that is used to transfer some hydrogen or proton ions from the matrix of the mitochondria to the intramembranous space. Now, this mitochondrial membrane. So from here to here, hydrogens are pumped, protons are pumped, and there is a pumping from each and every complex except some that we'll discuss in the next video. So there is pumping, right? Hydrogen proton ions get accumulated in the intramembranous space till what limit? Till the limit that they are eligible for passive transport back into the matrix, right? And that will happen only through a particular protein channel present here. And that, my friends, is the complex 5 ATP synthase, right? So basically this, let us assume this is a channel. So this is intramembranous space and this is the matrix of mitochondria. So whenever there is a gradient developed in here intramembranous space, then protons ion will again be directing themselves to into the matrix to maintain the gradient. So whenever this happens, there is a protein at jets here, like the turbine you see here, and there is a configuration change, conformation change in the protein structure, and that results in the generation of ATP. So yes, this was all for today's lecture. So I guess this series will be of approx 3 lectures. In 3 lectures we shall try to cover entire biological oxidation, not entire but main concepts. So do like, share and subscribe. As you like, share and subscribe the channel, we get the motivation. We need motivation to function as human beings, right? You know the effect of motivation in the physiological vivas. So do subscribe the channel, share this video to your friends. Stay tuned for next video. Thanks for watching. Stay tuned. Bye.